Most people today live to the age of 81, and what if you could cure all cancer? What if cancer didn't exist anymore? And you might think that would give us 10, 20 extra years. It doesn't. It gives us 4 years. Like, it's really a small, and that's because most cancers, again, are happening to people who are already quite old. And if it's not cancer, it's heart disease, it's something else. It's this whole constellation of things that happen as you age. So similar numbers, slightly better. If we could cure all heart disease, if every therapy was like curative that we were developing in the clinic today, that would help a little bit more, but not much. And the bottom line here is, what if we could, in humans, already slow aging to the extent that we've been able to over and over again in mice? What does that map look like? And it's amazing, right? Like, now we're looking at decades more. And importantly, too, these are not like sick decades, right? Like, here, if you're trying to sort of treat people that already have established cancer or heart disease, that's not like healthy lifespan necessarily, right? But if you intervene earlier, as again we've been able to do in mammals, that could translate to something much better. So this was really what inspired me to start what we're doing. And where we are today, and I think for those of you who are just getting into the science, I think a lot of outsiders don't appreciate how very new the science is, right? A lot of it actually that's happening today happens right here at the Buck Institute. But this is kind of a graph of showing the number of papers through time. And it's fair to say that until the 1990s, people thought, oh, aging is really hard. There's all these different things that go wrong as you get older. Why would you expect you could make one simple change and actually affect aging? And that idea changed in the early 1990s with the work of Cynthia Kenyon, Gary Revkun, et cetera, where they showed that you could delete one gene from a worm and double its lifespan, which I think was amazing, right? So that really, after that discovery, a lot of really great scientists entered the field, made some wonderful discoveries in C. elegans, the worm, in yeast and flies. And so there's, today, I think several hundred different things you can do to an invertebrate to make it live longer. So that's another important point, right? The number of interventions that work is not going to be too small. It's really only the last 15 years or so where there's been a growing list of things that reproducibly extend mouse lifespan. So this is the sort of where a lot of the research is focused right now. And these are things like senescent cell ablation, rapamycin, et cetera.
How long is the longest living mouse? If you do all of these things.
That's a great question. I don't know if they've actually been used additively. Yeah, do you know that, Eric? If we add like,
I think it's a doubling of lifespan.
Well, even with mice, have they gotten that far? Maybe like 40% or so, right? Genetically, they can double. Yeah, 2x. Yeah.
So what would I have to do to myself genetically to double myself?
Is it IGF?
Every single cell in your body.
First to solve, the delivery problem for gene therapy. That's problem one.
But the maximum with the drug is 30%.
To be fair, though, few have been tried so far, right? Like, just to reinforce the point that this is like a new field, an exciting field, in the last 2 decades, the National Cancer Institute has tested on the order of 20,000 compounds to see if they, make mouse tumors disappear. And in the same span of time, the aging field has only tested a few dozen drugs. So like, there's a couple of orders of magnitude off there. It's hard because the mice live long, right? So and it's hard because the field is not funded enough, right? So I think there needs to be a lot more basic scientific funding as well. Anyway, moving forward, the next layer, the most important layer, is humans, right? Because again, we're very different from mice. Mice in the lab are certainly much closer to us than a fly or a worm. But remember, like, mice never get Alzheimer's. Mice never die of heart disease. It's just not a bottleneck to their survival. They pretty much all die of cancer. So how translational are all these great discoveries going to actually be? An important point is that we already know today that humans can live a lot longer. And the reason we know that is because there's a lot of examples out there of people who make it to 110, 117. And in a lot of these people, too, everything still works, right? You don't want longevity without in poor function. You want longevity and also health span. One of my favorite examples on the right there is Robert Marchand. He was a competitive cyclist at age 105. Like, that's amazing. And of course, on the other end of it, there are still a lot of people who don't make it past their 60s, 70s. And so our whole approach at BioAge is to leverage this natural human variation to learn successful, safe ways that humans can live longer. And so this is just showing the distribution at age of death in one of our human cohorts. So our whole approach to discovery at BioAge is to run the equivalent of a 50-year experiment. And I say that it's a 50-year experiment because we've partnered with some very, very special biobanks that started collecting samples from healthy humans when they were middle-aged 50 years ago, and then followed them up for the entire rest of their lives. And I think this is really important for our approach. Like, humans age on the scale of decades. You really need molecular data that span decades to understand human aging. And our whole approach is to go into these like really rare, precious, banked human samples and interrogate them with modern technologies to see which pathways explain the difference between people who age poorly and age well. And I'm having another slide display problem where it looks beautiful here and bad there. But briefly, we take a multi-omics approach and collect data over the lifetime. So today at BioAge, we have over 65 million molecular data points, over 10,000 patients, over 45 years of aging. And the idea is that we have these sort of blood samples collected longitudinally through time over all of aging. And you can use technologies you might have hear about on this trip, like the proteome, the metabolome, the transcriptome. I essentially just making a big list of all the molecules in those samples, and then interrogating this data set and saying, what changes with aging, but even more importantly, like what predicts the future? Like what pathways, what proteins differentiate those 50-year-olds who go on to live a long, healthy life from those who don't? And I want to emphasize too that the biobanks we work with, they're not just for aging, they're also really, really valuable for health span, which is so important. So the patients here, they have these really rich electronic health records over the decades connected to these samples, looking at things like cognitive aging and cognitive function over the lifespan, muscle-relevant variables like walking speed and grip strength and how those change through time, other deep phenotypes. And just to show you like an aggregate signal, like we're basically looking in these sample sets, and this is an example of the proteome. So we're looking at 7,000 proteins circulated in human blood. And we're asking the question, what predicts like longevity, how long you live 40 years in the future, right? And you can see there's a lot of really interesting signals here with strong predictive power for how long you live. And interestingly, you can see here we've colored them basically by whether they're also associated to health span variables. So red for grip strength, green for cognitive decline, and some other factors here too. And so not everything that's relevant for longevity is also going to be protective for these other indications, right? So we're really most interested in those pathways and those proteins that promote healthy longevity. And the way that we think of our platform is that, as I said in the introduction, like we actually know a lot about aging today. There's been decades of scientific research. We have a growing list of things that work over and over again when you test them in animals. But which of those are also going to matter in human? So we think of this as like really the human layer. And if you look at our data set, which we visualized here as a network in the background, you can see that it's useful for two different kinds of activities, right? One, it can tell us which things we already know about can translate to human. Like, imagine if there's another gene that makes worms live 20% longer, and it's I'm sure it would make a great Nature paper. But the question is, does it actually matter for us, right? And if I also see a really big signal at the ortholog, the human version of that gene, well, now I'm interested, right? So it's a great sort of layer on what we know about animal aging. And two, because we age differently from other animals, there are going to be factors and proteins that really only matter to our aging, and we need this kind of data set to find them in the first place. And I think there's a growing constellation of really exciting companies doing important work in this area, especially some of the newer ones like reprogramming gene and cell therapies, xenotransplanted organs. And we see these different innovations as really paying off over different timescales. And our approach is really to benefit the patients of today with mechanisms that we can test in clinical trials in the near term. And I think that these will also all be additive together really over time. So at BioAge, we're focused on 3 different thematic areas of aging where we think there's high levels of unmet need, where we also as a company have a strong strategic position. Those areas are immune aging, muscle aging, and brain aging. And they're for the reasons on the right here. One is that there's a lot of information relevant to this in our human cohorts, and that's where all our insights come from. 2 is that there's a lot of opportunity in terms of the diseases and clinical trials that we can conduct. And 3 is that we have to believe the preclinical models, right? As I mentioned earlier, I think brain aging is so important, but mice never get Alzheimer's, and that's why we've cured Alzheimer's 100 times in a mouse and never in a human, right? So at BioAge, we restrict ourselves to those diseases that mice also get when they're old and in the same way. And if you compare us to others in the aging space, there's sort of really two major differences. One is that we're human first rather than animal model first. But two, which I also think is really important, is that we're just generating these giant data sets and asking the data what's the most important. And what that means is that we're going to be able to make lots of different mechanistic bets on completely different pathways that are uncorrelated to each other. So we're not just limited to one particular aging mechanism where we have all our eggs in one basket. And all of us, Jen mentioned this during her talk too, right? So great, you've got a drug, you believe it's an aging drug, now you have to do a clinical trial to prove that it works. How on earth do you do that? Because really, a clinical trial, if you actually want to measure aging, it would probably require thousands of people and like a decade or two of runtime, and it's just not feasible, right? So this is how our strategy at BioAge, and the important insight here is that most, not all, but most things that slow aging are also going to be curative for certain diseases, right? So we always select, at BioAge, the bets we've made, which is 3 programs so far and more on the way, are for mechanisms and drugs where there's a big aging indication at the end of the road, something like frailty or immune aging, where there's a high prevalence, that's the big mountain, huge market potential and high unmet need. But along the way, there's an initial model indication, a first practical disease, where we can run a really efficient clinical trial, get that first human de-risking signal, and build from there. And a great example, of course, is you guys are familiar with statins, right? Like today, statins are prescribed as though they were an aging drug. If you're over 40, you have some risk biomarkers, you get a statin. But they weren't approved first that way. They were first approved for an orphan indication. So a very narrow disease. And then the label was expanded over time. And that's how we at BioAge and many other companies in this space are approaching this challenge. And just briefly, I have a really great team. I love them. The company was founded by myself, my friend Eric Morgan, who is formerly an assistant professor of medicine at the University of Toronto. Paul Rubin is our chief medical officer, very experienced. We're really lucky to have him. He was formerly VP of Global Clinical Development at GSK and at Abbott before going into small biotech, and has personally overseen more than 10 drugs from preclinical through to approval. Ann Neal's our chief development officer, Dov Goldstein, our CFO, Peng Liang, our CBO, and a really great board. And really, so we're starting with these giant data sets, but that's really just step one, right? So we built the rest of the organizations that can continue to advance assets at scale from discovery through preclinical target validation through, again, to clinical development. So as I mentioned, yeah, go ahead, Peter.
The giant data set that you're starting with, a 50-year-old, again, how many different?
10,000 people,
10,000.
3 different ethnicities, yeah.
And have you actually run, so what do you get from them? Do you get cells? Do you get
It's blood.
plasma?
Because blood is pretty much all they're going to collect when you're middle-aged and healthy, right? Which is what I should mention too, we specifically chose people who, at baseline were healthy. So we don't want to detect signatures of disease. We want to see what actually predicts aging itself. Yeah.
So you have 10,000 blood samples,
and multiple time points. So yeah.
And I'm curious, how much blood did they actually give you?
How much blood?
Do they give you like a sample, like you get like a cc that you get to use over the course of the
Oh yeah. So these are biobanks that started 50 years ago, right? And they've got like a few mils usually, and it's a precious resource. So you have to. Usually you only need on the order of tens of microliters to do one of these newfound, whether it's proteomics or metabolomics.
So have you run all 10,000 already?
Oh, correct. Yeah. So all 10,000, and for some of them, multiple time points as well. Yeah.
And so, and were there any particular signals that lit up super bright? I mean, how many candidates do you have that you're excited about?
Yeah. I mean, like there's a lot, right? Like there's a lot of different. I think there's a lot of different levers on aging. Like again, I think there's only a few things that we've discovered that work well in mice, but we've tried so very few, right? So they're on the order of dozens, basically. And the way that we approach that at BioAge is we're prosecuting on those targets in the, with the sort of like the easiest ones first, But there's a long list, I think, of molecules we can work on.
Dr. Yamanaka told us last night, he started with the 20,000 genes. He picked 24, and it happened on 4 that actually worked. I wish you the same luck.
That's right in a way, though, right? Because I think it's really important to try out, have multiple different clinical bets, right? We just saw that with Unity today, actually,
Yes,
right?
exactly.
Their first one did not work. Their second one did. I think it's important that this field have a lot of bets. Yeah.
Do your samples have men and women?
They do have men and women, correct? And we favor things that are conserved. We're looking for things that are conserved first, for sort of the big roaring hits, basically. Yeah. I'll show you. I'm going to walk you through actually an example of one of our clinical programs later. So, yeah. I did want to mention in the middle, like we start with all this fancy data science, but then like that's really just correlational. I mean, there's some very good stuff you can do right now to sort of prioritize targets where we think this is causal, but you still need that step where you actually take an animal model, you intervene with the target, you see if you fix the disease, you see if they're aging better. So we've invested a lot in that at BioAge too, and we have over 5,000 old mice in our lab. As a biotech in this space, again, because it's such a new space, it's actually hard to buy enough old animals, so we decided to build that out ourselves, that capacity. And then similarly, we move forward into clinical development for the programs that excite us the most. And I'm going to basically walk you through an example for the drug that we are working on the most recently. So basically, we started off looking at our cohorts asking, what are the most important signals for muscle aging? And as I mentioned earlier, there's a lot of signals through time in our cohort that are relevant to muscle aging, things like grip strength, things like walking speed, lifespan itself. We put those together into like a composite muscle aging score. And from the bottom left here, you can just do a whole like histogram, like just to list all the proteins by like their muscle aging score. And one of the outliers there is a protein called apelin. And in fact, it's the most outlying protein that already had a drug available. Amgen was working on a drug that upregulated apelin that was in phase 1 studies. So we got really excited about that because we could take this great drug and test to see if it had a positive effect on muscle. I'm jumping the gun here a bit, but so if you go and you zoom in on this protein in our cohort, you see it as a nice linear relationship with health span as you would hope to see. So on the bottom right there, we're looking at all our middle-aged people, and as you can see there, the more apelin you have when you're middle-aged, the longer you live, the better your grip strength, the better your cognitive function as well.
Wow.
Yeah, so this is one that we really like. We're really excited about this one. Yeah.
And
George, can we test my
testing
insulin?
testing for apelin is just a blood test?
It's just a blood. Well, yeah, that's a good question. So you can do it on a research chip where you're also measuring 7,000 other proteins. I don't know if you go down to LabCorp if they even measure this, right? So it's too new.
What does
Yeah.
insulin do?
Lots of different things, like every aging target. So one of the muscle-specific things that we think is relevant, and where we've done some work, is it seems to improve the differentiation capacity of muscle stem cells. So yeah. And so just super briefly, you can look at.
So you're saying it allows you to build muscle easier?
So it also protects muscle from atrophy. So it's not, and it also has a role, like it's one of these things that's globally expressed as well, like it's also in the brain. We also see these cognitive function links. I don't know what it's doing there. It's not a muscle story there. So that's part of the story, yes. Yeah, yeah. So yeah, very briefly, I'll run out of time otherwise, but we looked at, the apelin network as a very strong, predictive power for how long you live. And, once we saw this in silico data, we got really excited. We reached out to Amgen. They gave us a bunch of their drug under an MTA. And we put it into multiple different models of old mouse muscle aging. And I'm just showing one piece of data here because this is the one that directly informed our clinical study. This is the one where we saw the biggest effect size in the smallest amount of time. And what this experiment is, it's really simple. You take an old mouse, you put a cast on one of its arms, and after 3 weeks, you take off the cast, and the muscle is atrophied a lot. You can cut it out and you can weigh it. And as you can see in the picture there, it's lost about half of its weight. And the mice who were on this drug did not show a significant decrease in muscle loss. So we're very excited by this finding that it could protect older muscle from atrophy.
Wow.
And that's how we're going to design our clinical trial. So.
If you were to create something that was organic, is there a way to create an organic product, or does it have to be a drug-related pharmaceutical?
So apelin is a peptide that circulates naturally in your blood. You wouldn't want to use the peptide itself as a drug because it has a half-life of 5 minutes. You'd have to be infusing it with IV continuously, basically. So it really is something where it's better suited to like a small molecule drug. Yeah.
How much would that cost to create?
How much it costs to create?
Yeah, like research and the timing.
Yeah,
Money.
that's a great question. I mean, these are all large efforts, right? Like, so this was a drug that Amgen had already built and had taken to phase 1 studies, but it had taken them, several years and dozens of chemists to get to that point. So it's not cheap. Yeah, it's on the order of, like, the cheapest you can probably do something like that is on the order of $10 million. So yeah.
Just a quick, would you mind?
Yeah, we're just looking at this study, this paper highlighting the effects of L-carnitine on the apelin receptor and its effects of actually increasing the activity of apelin on the receptor, just like my studies.
Yeah, that's interesting. There could be other. So apelin was actually first described too as an exerkine. So if you go out and exercise, it's one of these peptides that your body naturally upregulates, right? So that's at least partly related to the story as well. Yeah. But basically, we got excited by these data. We, under partnership with Amgen, we brought in the drug in-house. It's our drug now. And we're right now doing a phase 1b clinical trial with older volunteers. And it's really sort of as close as we could do to the mouse study. So we have people over 65, and they're going to be at bed rest, so sort of stationary for 10 days in bed. And after that, you see muscle mass decrease, you see a lot of protein changes in that time. And we're going to see if this drug can protect them from those changes.
Cut out their muscle.
No, we're going to image it. Something a little bit less invasive with the humans. but yeah, but still translational. That's right. And that's just really the first proof of concept, right? And then the indication, and just very briefly, we're going to look at a whole bunch of fun stuff in these people: muscle function, muscle size, muscle mass, as well as biomarkers and proteomics. And then from there, we're going to go to, as I mentioned, right? We want to go to practical indications first, and then like really big blue sky ones. So we're going to start with something called ICU diaphragm atrophy. So this affects basically the around 5 million people a year who are mechanically ventilated. And when you get put on a ventilator, your diaphragm muscle is used to working all the time. So it can actually atrophy in a matter of days. And if that happens, it's very highly predictive for days in ICU, for survival. And this drug, we're going to look to see in this patient population if it can prevent that atrophy. Again, just like a muscle atrophy prevention, like in our preclinical models. So it's a very severe first disease where we can get a rapid readout on whether this drug is doing what we hope it does in the human. And then after that, right, like on the longer road is something like sarcopenia and frailty itself. And I didn't show that here, but in one of our other experiments, we gave this drug in the water to just old mice that were just naturally running on their wheels. And this actually preserved and increased their running wheel speed and their grid hang strength. So to longer term, this could be a sarcopenia frailty drug for a much broader indication. And this is the statin story I told you earlier, right? So just as a brief reminder, that was just sort of a walkthrough of how we're thinking through the data story, the preclinical development, and the clinical development for one of our programs, starting with ICU diaphragm atrophy and building from there. And that's really the kind of approach we want to take to all of our targets at BioAge. And I love this quote. Our aim should be to help our patients